Okay, thank God, Julia, you are here. Yes, yes. But it's live now. We are live now. Yes, yes. A very good afternoon to all of you. Yes, yes. So how are you all? Let me know, please. Thank God we are live now because yesterday basically. Are you getting my voice? Yes, yes. Very good afternoon to all of you. Are you getting my voice, or should I switch off the phone? Please, sorry. Should I switch off the fan? Please let me know. Please, please. My voice is, some noise is there. Okay, then I'll just switch off the fan, and then, or when the students will be there, I just switch off the fan during our presentation. I will switch off it, but for some time let it be there. So how are you all? And first of all, sorry for yesterday's inconvenience. I don't know how and why it happened. I just came from the university back by 5, and then we have a online. We have to live by 5:30. But suddenly what happened, I don't know. But the stream is showing offline and offline. So in the night, my brother just update all the softwares so that we will be here again. Okay, so how are you all? And Juveria, how is your practical today? So some students of your batch has a practical today, but till you are till you are there at 4 p.m. So thank you a lot for being there with me. Yes, yes, Floris, good afternoon to you. Good afternoon to all. And I think one student has messaged me the topics which are remaining 1, 2, 9 and 10. So we have to cover those topics also. But let me check what are the topics. So the number 1 is basic principles of MBBT. So we will discuss it in the upcoming days, not today, because today we have to go further with recombinant DNA technology. So today we have to go with recombinant DNA technology. So we will, basic principles of MBBT, we will discuss these topics in the upcoming days. Then we have cell culture techniques. Then we have cell culture techniques, and then which is left? Are you here? Everything is okay? Let me check it. Yes, yes, it's okay. Are you getting my voice? Yes, okay, good. So we have 1, 2, 9, 10. So we have topic basic principles of MBBT. Then we have cell culture technique. Then bioinformatics and computation biology. And the last is the recombinant DNA technology. So basically, I will discuss with all of you, or the lectures which I will take is basic principles of MBBT, and then cell culture techniques. Definitely I will take these topics, but the topic which is on day 10, that is bioinformatics and computation biology, basically it's not in my field, so I will try to deliver the lecture on the bioinformatics also, because at that moment of time I don't have any type of presentation or something on bioinformatics. So for this topic I will apologize, but yes, I will deliver the lectures on bioinformatics from 29th of October. From 29th of October I have a lecture series on bioinformatics. So if you are obviously there on my channel, so those techniques or the bioinformatics we will done on 29th or onwards from 29th. So are you getting my point? Because I don't have any type of PPT right now related to bioinformatics. From 29th onwards we have a special lecture of 1 hour only on bioinformatics. So I just suggest all of you to kindly be there, then you will get more knowledge regarding bioinformatics, because you have only 1 day or a 1 hour lecture on bioinformatics on this platform or on this MBBT. Yes, I will give. Definitely I will give. But instead of only for a 1 hour, I just suggest you to be there with me for 5 or 10 days, so that you will get much knowledge regarding it. So yes, Jeet, need not to worry. I will definitely do it. You have not to take any type of worries. But that's only my suggestion, because from 29th of October I have a full 10-day series on bioinformatics only. So instead of covering it on or for 1 hour or for half hour or just doing it in hodgepodge, instead of it we will discuss it in detail starting from 29th of October. If it is unsuitable for you for that time, then obviously or definitely I will discuss, but not in detail, but in only a little one, or just, but not in detail, but just to summarize the topic, which is bioinformatics. So it's my suggestion to all of you to be there on 29th of October. But we have basic principles of MBBT, which we will discuss tomorrow, that is on Thursday, and then we will discuss cell culture techniques on the very next day, on Friday. And then on Friday, or you have 2 days more. If you have any type of query or you want to know any another topic related to this MBBT program, kindly let me know. We will definitely cover those topics also. Not only these topics which are there in your syllabus, but if you want to know something else, or you have some another point in your mind which you want to know or you want to discuss, then kindly let me know. So Juveria and Jeet, if you are here, then if kindly chat with your friends and let me know if you want to discuss any other else or any other topic instead of these topics which you provides me now. Okay, so are you getting my point? Please let me know. Principles of MBBT. If you are getting my points, please let me know. I am saying these 10 topics are okay, but if you want to know something else, or if you have some important point or some important technique or topic which is very important in MBBT, but we have not covered it, so kindly let me know, because the syllabus is not made by me basically. So someone else, maybe your previous teacher, has made this topic. So I am not sure whether all the topics are covered. Maybe covered, of course, definitely may be covered. But if you have some any another topic in your mind or you want to know, then kindly let me know. No issues regarding it. I will take it up. The class will, like today is Wednesday, we will take it up to Saturday or Sunday also. There is not any issue regarding it. So that's only the thing I want to say. But only for bioinformatics, I suggest you to be there with me in the next series, because bioinformatics is a very broad topic. We will not able to cover it in half hour or 1 hour only. So that's my point. So are you getting my point? First of all, let me know. Are you getting my points? Kindly reply please. So we have Jeet here, we have Gayatri here, then we have Juveria. Yes, you all are here. Khan Maria is here. So are you getting my points? The thing which I want to convey to all of you, that we are not limited only to this 10-day series or only to this or the remaining days. No, it's not like that. You are most welcome all the time. if you have any query, if you have any topic in your mind, we will definitely discuss. So need not to worry that you have only few days left and then any topic will be left. No, it's not like that. You are not leaving me basically, or I am not leaving you. We have another series, or we have another sessions also, like in the evening also by 8 p.m., and then so and so on. There are so many series, so I welcome all of you to be there. If the content is according to your point of interest, then please learn those techniques, because I think you are paying something to for this lecture series maybe. So it's your right to learn more and more and more. Okay. So the viewers are very less. If Juveria or Jeet, kindly send the link within your personal groups or your friends group, so that they will also join. We will start our today's session. So if you are here, kindly share the link, because I don't know what happened yesterday. It's not the link is sharing. I am able to share the link, but it's showing offline and offline. Even I tried so many times, it's showing offline. I don't know why. Yes, Ria, good afternoon. How are you, Ria? Aditi, yes, Aditi is also here. How are you, Aditi? We have Devita here. Gayatri is here. Vatsal sir, is it Vatsal sir or some student is there? So then we have Ganesh Das is here. Gayatri is here. So I welcome all of you. Lekha is here. So kindly reply. Let's just interact with each other. Then we will start the session, so that other students will also join with all of us. So kindly reply if you are listening to me, please, and my voice is audible. Let me know this also. Yes, Jaguriti is here. How are you, Jaguriti? Prachi is also here. Prachi, how are you? Yes, we have so many students here. Good. So Jeet and Juveria, kindly share this link with your another friend, so that they also join with us. Bhagyashri is also here. Bhagyashri, how are you? How are you, Priya? Okay, Prachi says she is good. Okay, very good. Amita is also here. How are you, Amita? So I am just requesting you that we are not bounding for this 10-day series, or we are not here only for 10 days. You are always there with me. If you want to learn something new, or if you have some type of free time to learn something, then you must carry forward with our next series or another topic, so that you will get some knowledge also. Okay. So there is no issue anything regarding it. Knowledge is knowledge only, either in MBBT or in biochemistry or in anywhere. So you have to be there also. So have you any query, or you want to say something? Kindly let me know. Why are you not speaking today? What happens? Have you taken your lunch? Why are not you speaking? Have you any query, or you want to ask something? Kindly let me know. Or you are just preparing for your exams, that's why you are a little bit busy. Ask your questions also, please, because I am only who is questioning you and you are replying. Just ask you. Kindly share something if you have to. Yes. So Gayatri is saying, ma'am, can you please suggest us some reference books for the topics we have covered so far and will be doing in upcoming days? Yes, definitely. I will share some important books, not from India, from foreign authors, which are very important in these molecular biology techniques. Like we have a book of Gilbert, and then we have some another books, but I will share with you the proper link. I know this question. to share the books with you. Not the question, but the query. So we have so many of books these days available. I will send the link. So again, my my priority is to all of you that you have to remember the basic things first. You have to know the principle of every technique which we have done, either it's western blotting or southern, northern, or flow cytometry. If you are able to learn the basic points or the very first important principles, then there are so many versions of a technique, you are able to get those versions also. Okay, so are you getting my points? So Jeet, where are you? Jeet, are you here? So I don't know why are you not replying today. What happened? Everything is fine, or have you your exam? Or is it basically the noon session? That's why it may be a bit early for you also. Is it like that? Please let me know. Actually, because of yesterday we are not able to take the yesterday's class. That's why I think that start it by 4 p.m. because I have not to go anywhere today. So that's why. So please let me know why everything is fine. Hello. Everything is fine. Please let me know. Yes, yes, yes. Okay, okay, good, good. Everything is okay. Okay, good, 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 good. Okay, okay, okay. So why are not you asking anything, or you are not, because it is too, in the noon we take the session. Maybe this may be the reason also. Rituja, how are you? So just waiting for 2 or 5 minutes more for some students to join because some have practical today. Okay, okay, it's reaching me late. Okay. So Jiveri, I ask you how is your practical today? You don't reply me. I ask you how is your practical today? Okay, time slot is okay. Okay, thank you. Thank you, Rebita. And even I am asking Jeet, are you here? And he is not replying me. Okay, you did not have your practicals today. Okay, okay, okay. Good, good. Man, network issues. Network issues are always there. I don't know why. But in my side, the connection is good today. Even I am able to, you know, your messages are there within time. Okay, because of MBBT, you shifted your practical. Okay, okay, that's so nice of you. That's so nice. Thank you a lot, Juveria, and even your whole class. Not only you, but the whole class. I'm not know how many students or which one is there in your batch or in your class. So I want to thank all of you if you postpone your practicals because of this MBBT series. So that's so nice of you. Thank you all. So shall we start now, or we will wait for 2 minutes more? Or are you writing some type of review article or not? Because in the another session, the students in biochemistry, they are so much willing to know the review article. They are giving me so much of messages. So are you also doing some type of review articles or not? Kindly let me know. Are you also doing yes, 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 ma'am, we can start. Okay, okay, I'm just starting. So I'm just asking that either you are also writing review papers or not, because you didn't provide me any information regarding it. Okay, you started writing the paper, Vishakha. You started writing the review paper. So today we have to start our topic with recombinant DNA technology. All of you must have done it before also, maybe in your classes or somewhere. So let's start. So let me ask you. So is it visible to all of you? Vishakha, Siddhi, Devika, kindly reply. Hello. Are you getting my voice? No, actually I have just there is the PPT, but I came back into the YouTube studio. Just asking you the questions. So are you able to get the slides? Please let me know, and my voice is okay. Also let me know this. Okay, 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 okay. Okay, good, good. So now we will start our topic, which is recombinant DNA technology. So we have to know all these things, like basically what the recombinant DNA technology is and what's their procedure, and basically our main focus is on the applications which are there in the recombinant DNA technology these days. So what's recombinant DNA technology? DNA molecules that are extracted from different sources and chemically joined together. So what happened in case of DNA technology? The DNA molecules or a specific DNA segment is taken from a different source, and then this DNA segment is jointed in some another source. So similarly here, if you are able to see, the DNA molecules are extracted from different source. So the segments are taken from some another source and chemically joined together. For example, DNA comprising an animal gene may be recombined with the DNA from a bacteria. So an animal gene may be recombined. So what we do, we will use bacteria for, the small DNA segment is joined in the bacteria. So this is the recombinant DNA technology, the technology in which we are using the DNA segment from one source and then it inserted into the some another source or chemically joined somewhere else. So first of all, we all know that DNA was this, the structure of DNA, that is double helical structure like this one is came to know in 1953. Then in 1967 we have isolation of DNA ligase. The enzyme is there and we have isolation of that particular enzyme. Then we have isolation of RNAs. There must be an. So this is the isolation in 1917. So but in 1972 when we are able to understand the concept of DNA technology, and the person is Paul Berg, who generate or who introduced the term RNA or DNA technology. That's a recombinant DNA technology in the year of 1972. Then again one another scientist, that is Cohen and Bohr, in 1973 produced first plasmid vector capable of being replicated within a bacterial host. So we have a bacterial host. Host is what one who will receive the DNA segment in which we insert or join the DNA segment. So produced first plasmid vector capable of being replicated within a bacterial host. So this is the basically you can say the history of recombinant technology, that from 1953 onwards when we have a structure of DNA, the structure of DNA is discovered by the scientists. Then after from 1953 to 1972 we are able to generate the technology which is known as recombinant DNA technology. Now what are the goals of recombinant DNA technology? In other words, you also say what is the need of recombinant DNA technology? Why we need this technology? That is the recombinant DNA technology. Why we need this technique? Because the very important or the very first thing is isolation and characterization of a particular gene. Suppose if we want a particular gene, or if we want a particular DNA segment, we want to study only particular portion of a gene or of a DNA segment. For this purpose, for isolation and characterization, we need this technique, which is recombinant DNA technology. Then to make desired alterations in one or more isolated genes. Yes, this is another point. If like scientists always try to make changes, so if we want to make some type of changes or alterations in one or another genes, for this purpose we need a recombinant DNA technology. Then to return altered genes to living cells, artificially synthesize new genes, because in the era of this science or in the era of this fast technology, you can say, to artificially synthesize a new gene we need a technology. That how we will synthesize a gene which is helpful for curing any disease, or which is helpful for curing any type of cancer, or you can say any type of disease which is controlled by a specific gene segment. So if we are able to isolate or we are able to synthesize that particular segment which is curing any autoimmune disease, like there are so many autoimmune diseases which are not curable at all. You can't cure those diseases because they are autoimmune in nature. Your own body fight against yourself. So to cure those autoimmune diseases or Alzheimer's diseases, like these diseases which are related to our body health, to cure those diseases we need to synthesize the new gene, or we need to isolate or characterize only that particular gene which has a specific for a particular disease or for a particular reaction in our body. Then alternating the genome of an organism, understanding the hereditary disease. So this is the most important point in recombinant DNA technology, because by the 19th century we are able to know so many types of diseases, like we have diseases, basically the autoimmune diseases, in which our body fights with our own self, or the body cells fight with the own body. So to cure those types of diseases, there is a need of recombinant DNA technology. So understanding the hereditary diseases and their cure. So this is the important point, that is why we need the recombinant DNA technology. And improving human genome. So if the scientists are doing so much type of discoveries or research, basically only to improve the human race. So improving the human genome, for this purpose recombinant DNA technology is the must. So let me check whether you are getting it. Then I will move further. So are you getting it? Everything is fine? I think you are, maybe. So just reply if everything is okay. Devita, Vishakha, just reply me. Yes, okay, good. Priya, yes, good. So basically why we need the this technology, or you can say recombinant technology. So okay, you are getting it. Okay, good. Okay, good. Okay, good. Jeet, you are here. I asked you some question. I don't remember now, but in between the chat, I just forget the question now. Maybe I am asking about review paper. I don't remember, but I am asking you something. So it's okay. Now let's move. So this is the need of recombinant DNA technology. That's why we need the recombinant DNA technology. The very important thing is to know the hereditary diseases which are incurable. So for this we have the recombinant DNA technology to know the hereditary disease, or also to isolate and characterize a particular segment of a gene. We don't want whole genome, but we want only a particular segment. For this purpose we need the rDNA technology. Now the another point is basic principles of this technology. The very basic principles, that how this technology works. So number 1 point is generation of DNA fragments and selection of desired piece of DNA. So first of all we have to know which segment we want to take, the DNA segment which we want to take. Then insertion of the selected DNA into a cloning vector to create rDNA or chimeric DNA. Then what will happen? Suppose this is a DNA segment, suppose like this one, and this region is our desired region, like this region is controlling, or this gene is controlling AIDS, suppose, or the HIV virus, suppose. So we want to study or to isolate this particular region. So first of all, the insertion of the selected DNA into a cloning vector to create rDNA. So this particular segment is inserted into the cloning vector to create rDNA or chimeric DNA. Then introduction of the recombinant vector into the host cell. Then the recombinant vector, which consists of the particular DNA segment, is introduced into the host cell. Multiplication and selection of clones containing the recombinant molecules. When we found a perfect host cell, then what will happen? There is a multiplication of the recombinant molecule which is just attached with the cloning vector. Then at the end we have expression of the gene to produce the desired product. So first of all, isolation of the DNA, the particular DNA segment, then cutting of the DNA, and then cloning of the joining of the DNA along with the cloning vector, and the last is the amplifying or amplification or the multiplication of the desired segment. So these are the four basic points which are there in the recombinant DNA technology. We have isolation of particular DNA segment. Number 2, cutting of that particular segment which we want, then joining it with the cloning vector, and then amplification in the host cell. So these are the 4 points in case of recombinant DNA technology. So the number 1 point is the isolation of the DNA. So first of all, like we know DNA isolation, protein isolation and RNA isolation. So we have to isolate a particular DNA segment of our point of interest. So these are the cells which are lysed with the help of detergent to break the plasma membrane. Then the cells are there, they are treated with RNAs, so to destroy the DNA. Suppose in case of RNAs we just add DNAs, what will happen? Instead of RNAs we just add DNAs, what will happen? The DNA which we want will be digested or will be destroyed by the DNAs. So instead of DNAs we have to add RNAs which destroy the RNA. Then cell debris is pelleted in a centrifuge. The supernatant containing the DNA is transferred to a clean tube. So this supernatant is transferred to we have to leave this pellet and we have to take the supernatant or the upper solution or the above solution. The DNA is then precipitated with ethanol, and this DNA which is of our desire or which we want to isolate will move further for the next step, which is the cutting of the DNA. So DNA can be cut into large fragments by mechanical shearing. Restriction enzymes are the seizers of these molecular genetics. So we have this genomic DNA, like this double stranded genomic DNA. We have base pairs here, AT, GC, GC, AA. Similarly here against this complementary base pair. So we have to use the restriction enzymes which will break a particular segment of our point of interest. So restriction enzymes are there which act as a seizure. So what are restriction enzymes? A special class of sequence specific enzymes found in basically in bacteria. So what these restriction enzymes will do, they cut the DNA segment at a particular site. So restriction enzymes recognize DNA based sequences that are palindromic in nature. So what are palindromic in nature? Just reply, what are palindromic sequences? You must know about it. So what these enzymes will do, they cut the complementary base sequences for easy circularization. So what will happen, the complementary sequences will be, so this DNA sequence is there, so we have cut it here, from here. So they will be breaked or they will become the different segments. So this is again, we have this restriction enzyme which will help us to break the DNA fragments. So this is the cutting of the DNA, in which we will cut the segments according to our point of interest. And after it, what will happen? Now there is a breaking of this sequence like C, T, T, A, and from here we will get another sequence, and both this sequence and this sequence will form a complementary base pair. They will form a bond or they will pair with each other, and they are joined by the enzyme which is DNA ligase. So they are joined by the enzyme DNA ligase. So this is the second step, which is the cutting of the DNA. So this is the thing, we have DNA, but DNA is present in the nucleus or in the mitochondria also. So but here in this case the DNA is present inside the nucleus. So we have to extract and digest it. So number one step is the isolation of DNA, and we will get the DNA. Now this DNA fragments, what we do, ligated to the vector. So by cutting the specific DNA which we want, which is our point of interest, this this particular segment is then ligated into a vector. So this is the insertion of the segment, and after it we have to introduce it into the genome cells. So these vectors or cloning vectors, they are introduced into the genome cells. So if you are able to see, the purple one is here and the red one and the green one. So these are introduced into the host cell. So this is the whole process of the recombinant DNA technology basically. This is again the example of recombinant DNA using a human gene and a bacterial plasmid. So first of all, what we do, like this is the insulin gene, the gene which is for the your diabetes, which is for the diabetes, insulin gene is there. So this gene is our point of interest. So first of all, with the help of restriction enzymes, we have to cut this. So restriction enzymes cleave insulin from the chromosome and creates corresponding gap in bacterial plasmid. So these restriction enzymes, what they do, they will cut at a specific segment. Then cleaved insulin gene is placed with cleaved plasmid. So what will now they are doing? They, with the help of DNA ligase, introduced this insulin gene into the bacterial plasmid. So if you are able to understand that this insulin gene is introduced into this thing, that the gene is insulated into the plasmid or a vector. So it is introduced into the plasmid with the help of enzyme DNA ligase. Now this red one or this insulin gene is then further transferred into the host cell. So DNA ligase joins the sticky end of the insulin gene and plasmid DNA. The transformed plasmid is inserted into a bacterial cell. So this red gene, which is our point of interest, or you can say insulin gene, then passed into the bacterial cells for further, you can say for further multiplication or for further cloning. So this is the basic principle of recombinant DNA technology. We have number one step, which is isolation, then we have cutting, and then we have insertion into the plasmid, like this plasmid, and then we have final insertion into the host cells like bacteria. So the transformed plasmid is inserted into the bacterial cell for a multiplication basically. So these are the restriction endonucleases, these are the enzymes. These enzymes are for the manipulation of DNA, are bacterial enzymes that can cut or split DNA at specific sites. So endonucleases cut the segment of a DNA at a particular point. They were first discovered in E. coli, restricting the replication of bacteriophages by cutting the viral DNA. Thus, the enzymes that restrict the viral replication are known as restriction enzymes or restriction endonucleases. So, restriction enzymes are also known as restriction endonucleases. Then we have, when we have done the, you can say by the use of enzyme, we just cut the segments, or you can say by the help of restriction enzymes. Then we have the another thing, which is the recognition of the sequences. So recognition sequences is the site where the DNA is cut by a restriction endonuclease. Restriction endonucleases can specifically recognize DNA with a particular sequence of 4 to 8 nucleotides and cleave. So restriction endonucleases, which we have done now, they will cleave a particular sequence. And yes, the other important thing is that they will cut either at the sticky ends or blunt ends. If you are able to see here, so they have the sticky ends. We have two types of ends, either the sticky ends or the blunt ends. So this is done by the restriction enzymes or restriction endonucleases. So DNA fragments with sticky ends are particularly useful in this technology, since single-stranded sticky DNA ends can easily pair with any other DNA fragment having complementary. So generally in case of our DNA technology, we have sticky ends, that is the cut is in between the DNA segment. So these are the points in case of you can say the recombinant DNA technology. Then we have the types of restriction enzymes. Type 1 restriction enzymes 1, type 2 and type 3. So we will discuss it, but let me know, are you getting my points? So is it everything is okay? Yes, Julia is here. She is saying, ma'am, palindromic sequences are sequences which are same from both ends. Very good, very good. So only one student reply. Where are the others one? Why are not you replying? So are you getting my point? Kindly let me know. Yes, yes. Are you tired today? You are not. I'm not able to see the enthusiasm in all of you today. So are you getting my point? Please let me know. Please, please, please. Kindly reply. Yes, yes, Manpreet is here. Yes, yes. Okay, no. Jeet say no. What happened to you, Jeet? Why you are saying no? Okay, okay. You all are good. Okay. I think maybe you are tired today or something like that. Okay, 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 okay. Good. Amita is also here. So okay, now I will move forward. So this is the thing which I want to know, whether you are getting it or either you are here or you just okay. So you all are good. Okay, so now next. So I have said that we have three types of restriction enzymes. We come to know what are the restriction enzymes are. That they cut at a specific sequence. They cut either sticky ends or blunt ends. But in case of DNA or recombinant DNA technology, either sticky ends or blunt end are there, but generally sticky ends, in which the cut is in between the segment, is used for this technology. So this is the important thing. Then we have types of restriction enzymes which we have. We have type 1, type 2 and type 3 restriction enzymes. So what are the type 1 restriction enzymes? They do not generate specific fragments, require the presence of magnesium. So they do not generate specific fragments. That means the particular fragment which we want, they do not generate it. Okay, so these are the number 1 or type 1 restriction enzymes. So require the presence of magnesium, ATP and adenyl methionine, which activates the enzyme. So even it needs adenyl methionine to activate. So this type 1 enzyme is activated with the help of the say. So that activates the enzyme, tracks along the DNA for a variable distance before breaking. So these restriction enzymes have little bit of disadvantages, that they do not cut the specific segment or specific fragment. Then we have type 2. They cut within or immediately adjacent to target sequences. So they are nearby the target sequences, where the sequence which we needed. Generate specific fragments. Magnesium is essentially for it. No ATP is required and no adenyl methionine is required. So they are little bit advanced from the type 1. Then we have type 3. They require ATP and adenyl methionine for cleavage, make breaks in DNA 25 base pair away from the recognition site. So they require ATP and also the as adenyl methionine for cleavage, but the main point is they breaks in DNA 25 base pair away from the recognition sequence, or you can say they are very near to the sequence recognition sequence which is needed. So these are the enzymes, like we have VAMH1, EcoR1, HIND2, 3. These are the very important, you can say, the restriction enzymes, and these are the microorganisms on which they work. So if you are able to see this BAMH1 able to cleave between this base pairs, or like from here they cleave the DNA fragment. Similarly here there is a cleavage. Here also there is a cleavage. So these are the like bacillus bacteria, we have Escherichia coli, we have Haemophilus aegyptus, so or you can say Streptomyces. So these all are the microorganisms, and you have to remember the name of the enzymes: BMH1, HIND2. So you have to remember the name of the enzymes also. So this is a restriction enzyme action of EcoR1. If you are able to see, these are the enzymes, this one and this one. So they break at a particular sequence, like here you are able to see. So they break the particular sequence in both the cases. Then we have to join the different DNA fragments, and this where there is a joining of different DNA fragments, this area is known as recombinant DNA. So this area where there is a joining of the sticky ends of different DNA segments, this area is known as recombinant DNA. Okay, so this is again, first of all we have to, this is the case of RNA. So first we have collected the RNA, then with the help of reverse transcriptase enzyme we will get DNA. Like from DNA, with the help of transcription we are able to get RNA. Okay, but from RNA we get cDNA with the help of reverse transcriptase. So this cDNA is inserted into the plasmid. This plasmid is then inserted into a bacteria, and then we are able to get the cDNA library. So first of all the segment is inserted into the plasmid, and then this plasmid with the help of restriction enzymes, the DNA segment is inserted into the plasmid, then this plasmid is then moved further to the, you can say, the bacteria. So this is the whole procedure of recombinant DNA technology from DNA. Like this is from the DNA as a source, like this is from the DNA. So here the everything is with the help of DNA, but here in this case this is from the RNA. So from RNA we can also collect the cDNA. So this is from the mRNA, and we are able to get the cDNA with the help of enzyme reverse transcriptase. So this is the again the thing, we have the enzyme which is EcoR1. So we have breaking here, then the two sticky ends will join, and we will able to form the the mixture of the DNA fragments, or different DNA fragments, or you can say the desired DNA fragment get inserted into the plasmid. So these are the blunt ends. Blunt ends are what? No further cleavage. Like this is the proper breaking from this to this, or from any, or like here they are saying from 3. So restriction endonuclease plate producing the blunt ends which have no further cleavage or cutting. And then we have the sticky ends. I think there must be sticky ends also. So DNA ligases, now we have the restriction enzymes or restriction endonucleases to create to break or to cut the DNA fragment. Then we have to join two different DNA fragments with the help of an enzyme which is DNA ligases. So these are originally isolated from viruses, also occur in E. coli and eukaryotic cells. So basically they are found in viruses. The cut DNA fragments are covalently joined together by DNA ligases enzyme. So with the help of DNA ligases we are able to join the segments. DNA ligases join the DNA fragments by forming a phosphodiester bond between the phosphate group of 5 carbon of the 1-deoxyribose with hydroxyl group of the 3rd carbon of another deoxyribose. So basically DNA ligase is the one enzyme which is able to join together the different DNA segments. That is why it's known as DNA ligases. So what will happen after? So these are DNA ligases enzymes. So there is a joining of the DNA. If you are able to see here, we have the particular enzyme which is EcoR1 endonuclease, which will break the particular segment, and here there is an introduction of the desired DNA segment which we want, and there is a joining of these two segments with the help of DNA ligase enzyme. So with the help of DNA ligase enzyme, we are able to join the two different segments of our DNA in the case of a vector or a plasmid vector basically. So this is a joining. Then we have to do amplification. Now what's amplification? Amplification is a multiplication of the desired DNA segment which is inserted into the plasmid or a vector. So transforming the recombinant DNA into bacterial host strain, the cells are treated with calcium chloride, DNA is added, cells are heat shocked, DNA goes into a cell by somewhat unknown mechanisms. So once in a cell, the recombinant DNA will be replicated. So within the, you have to remember on which host it will go inside the bacterial host. So the recombinant DNA which is present in the vector, or you can say in the plasmid, then it is to be transferred to the bacterial host. The host is the bacterial. So when the cell divides, the replicated recombinant molecules go to both daughter cells, which themselves will divide later. Thus DNA is amplified. So DNA gets amplified. So this is the amplification. This is the amplification. So first of all, this sequence is enter or just put inside the vector, and then this sequence will further put inside the host cell, which is basically the bacterial cell. So these are the enzymes which are used in the recombinant technology. We have DNA ligase enzyme which helps in binding of different DNA molecules. We have restriction endonuclease enzyme which cuts at a specific site. We have DNA reverse transcriptase which makes the copies of DNA from RNA, RNA polymerase enzyme, exonuclease enzyme, and alkaline phosphatase. These all are enzymes which are used in recombinant DNA technology. So are you able to get it till now? Let me check please. Are you able to get it? Signal is okay, or is there any type of buffering? Are you getting my point, students? Then I will ask you one question. Okay, Shaka is saying yes ma'am, we are getting you. Okay, good. So I will ask you, may I ask you one question now, which you have to give me answer, not today, not right now. You will take your time. You will take your time and give me answer even on tomorrow also. So this is a question you have to remember and have to tell me in the next session, or maybe now also. Like in case of recombinant DNA technology, we are saying that we are amplifying a particular DNA segment. We are saying that we are amplifying a particular DNA segment of our interest. First, this segment is inserted into the plasmid vector, and then it is inserted into the host cell, where there is a multiplication of a particular DNA fragment. This is the case now, that we are getting a particular segment. Now my question is that we have another technology which we have done the day before, which is PCR, that is polymerase chain reaction, in which similarly we have a particular DNA segment or a particular gene. By the help of PCR machine, we are able to amplify that particular region, like in the number of 2 to 4. I have shown you the diagrammatically or video in which there is a multiplication of the particular DNA segment, like 2 to 4, 4 to 8, 8 to 16. So within 30 minutes, how many types of, how many number of fragments we get, we also see this on the previous day. So my question is this, that if we are able to amplify a particular segment by using PCR, if we are able to amplify a particular segment in PCR also, we have extraction, we have DNA isolation first, then we take the particular DNA segment in the PCR tubes and we perform the PCR. So if amplification of particular segment is there in PCR, so why we need rDNA technology? Are you getting my point? why we need rDNA technology if PCR machine is there for the amplification of or for the increasing the numbers or the replicas or the clones of the particular DNA segment. Why do we need rDNA? So are you get my point, or should I repeat again? If you get it, then please take today's time. There is not any hurry. There is not any hurry. Take today's time and give me proper answer tomorrow. Okay. So are you getting my point? Okay, good. So if we have PCR technique is here for the amplification of a particular DNA segment, why do we need rDNA technology? Because same things are PCR and same things are almost same things are there in rDNA. So kindly take your time and please give me the answer on tomorrow's session. Okay. Okay. Now I just move further. So we have done what we have done. We have done the enzymes which are used in the recombinant DNA technology, DNA ligase, restriction endonuclease 1, reverse transcriptase, polymerase, and some other enzymes are also there. Now we will move. This is the whole mechanism or whole procedure of rDNA. Now we have to elaborate little more the specific thing, that is the DNA segment which we have done, the enzymes which are used we have done. Now the vectors or the plasmids in which we are inserted the particular DNA segment. So what are vectors basically? A vector is an area of a DNA that can join another DNA part without losing the limit of self-replication. So in vector, or you can say in plasmid, there is an insertion of a DNA segment. So should be capable of replicating in host cell. Should have they are just discussing the properties of a vector. So they are saying when they go inside the host cell, which is basically the bacteria, like this is the host cell, this is the bacteria, so plasmid is able to replicate or plasmid is able to multiply, and this plasmid is carrying our gene of interest. So should have convenient RE sites, that is restriction endonuclease sites, for inserting the DNA of insert. Should have a selectable marker to indicate which host cell received recombinant DNA molecule. Should be small and easy to isolate. So one thing is that it will enter into the host cell very easily. Then another thing is it should move out from the host cell also easily. So these are the characters of a vector which are there or which are required for rDNA technology. So these are the types of vectors which we basically use. Basically we use plasmid, but we have lambda phase, cosmid, and express science. So these are another type of, you can say, vectors which are used in rDNA. So this is the plasmid number 1, which is small circular DNA molecule that are separate from rest of the chromosome. So this is a very small DNA small plasmid. They replicate independently of the bacterial chromosome. So this is the very important point of the vectors which we are using, that they replicate independently. They don't need a proper host like a bacteria for replication. They replicate independently of the bacterial chromosome. If they are in the bacteria has its own structures, his own, you can say, chromosome or DNA, but these plasmids, they will replicate their own. They have no issues with the bacterial chromosome or bacterial, life cycle or something. They will replicate their own. So they are useful for cloning DNA inserts less than 20 kilobase pairs. So these plasmids are basically used for those DNA segments which are less than 20 kilobase pairs in size. So up to 20 kilobase pair DNA, this plasmid vector is suitable. Inserts larger than 20 kilobase pair are lost easily in the bacterial cells. So these plasmids will carry only the DNA up to 20 kilobase pair. And if you add the DNA segment more than 20 kilobase pair, what will happen? It will lose in the bacterial cell. So we have to take the segment up to 20 kilobase pair. So you have to remember this point, that we have to take the segment less than 20 kilobase pair in the in the plasmid vector. We need to insert the DNA less than 20 kilobase pair. If you take more than 20 kilobase pair, what will happen? The fragment of interest which we need gets loose or gets lost in the bacterial cell. Then we have plasmids. Then we have their own properties, another properties, that plasmids are extra chromosomal double-stranded circular DNA molecules. These plasmids contribute 0.5 to 5% of total DNA of bacteria. A few bacteria contain linear plasmids. So these are the properties of plasmids. Yes, the important property which we have to know. The plasmids carries genes resistance for ampicillin and tetracycline that serve as a marker for the identification of clones carrying the plasmids. This thing I will tell you later, but these are the properties of the plasmids which acts as a vector. So if you are able to see here, yes, the point that plasmids carries gene resistance for ampicillin and tetracycline that serve as a marker for the identification of clones. So if you can see here, this plasmid carries the genes for antibiotic resistance. This is the gene. This yellow one is the gene for antibiotics resistance. This is the DNA which we want to replicate, which we want in high numbers. So this DNA with the help of enzyme endonuclease comes inside the plasmid. Okay. Now what will happen? Here are two cases. Now what will happen? If this plasmid, the only reaction will take place, or only further reaction will take place when there is a the DNA which will be taken from the outside and this antibiotic resistance gene. Now what will happen? Only this plasmid will able to enter into the bacterial cell and do the multiplication. So this is the recombinant plasmid. The another plasmids which don't have this, you can say, DNA of interest, or there is any type of mismatch, they are not able to get any type of amplification, or you can say cloning. So this is the important point. Like here also, if you are able to see, these are the enzymes which are restriction endonucleases which will cut the segments, and this desired segment, this purple one, comes inside the plasmid, and now there is a replication of this recombinant DNA, but not this mismatch. So these mismatches will not be processed further, but only the recombinant DNA will be processed further. So only recombinant DNA, which consists of the plasmid along with the gene of interest, will be processed further. Sorry. So again here we can see this is a plasmid vector. We have specific cleavage sites on which the gene of interest will enter, and then this you can say plasmid will replicate into more numbers. So this is the number one, which is a plasmid vector. We have some another vectors also, but we will just discuss them. Lambda phase vectors. So lambda phase vectors are recombinant infections containing the phase chromosome in addition to the embedded outside DNA. So what they basically contain? Phase chromosomes. All in all, phase vectors can convey bigger DNA grouping than plasmid vectors. So in plasmid vectors, as we all know, the number is limited to the number or the size of DNA is only 20 kilobase pair. So if the size of DNA is more than 20 kilobase pair, if it is more than 20 kilobase pair, then we have to use this lambda phase. At that time, plasmid vector will fail. So for the bigger DNA or the larger molecules of DNA, we need lambda phase. Then we have cosmids. They are the vectors possessing the characteristics of both plasmid and bacteriophage. These carry larger fragments of DNA compared to plasmids. So cosmids are also able to carry larger size DNA. So cosmids are hybrids of phages and plasmids. So cosmid is basically what? It is a mixture of phase, that is bacteriophage, and plasmids that carry the DNA fragment of 45 kilobase pair. Like our first plasmid, our first vector, which is plasmid, carry only 20 kilobase pair DNA, but this cosmid will carry 45 kilobase pair DNA. That's why this is important. They can replicate like plasmids but can be packed like phage. So they have the properties of both, that is plasmids as well as bacteriophages. So this is the cosmid vector, which is important. Then we have expression vectors. What they are? They are the vectors that carry host signals that facilitate the transcription and translation of the inserted genes. So they also facilitate the transcription, that is from DNA to the formation of RNA, and translation, that is from RNA to protein. So they carry the those functions also. So they are useful for expressing eukaryotic genes in a bacteria. Basically, expression vectors in which we are able to do transcription and translation, they are generally used for the eukaryotic genes, or you can say the larger genes. Then we have yeast artificial chromosomes. They are also the vectors. Basically, they are the yeast vectors that have been engineered to contain a centromere, telomere, region of replication, and a selectable marker. So these are little bit more advanced, you can say, the vectors to carry the particular DNA segment. The only function they have, either we have cosmids, we have bacteriophages, we have plasmids, we have these yeast, is to carry the particular DNA segment of particular molecular weight or particular size. So they will carry up to 1000 kilobase pair. So one we have 20 kilobase pair, that is plasmid only, and these one will carry 1000 kilobase pair of DNA. So that's why they are more advanced, or they can be used more. So they are useful for cloning eukaryotic genes that contain introns. So they are used for eukaryotic cells or eukaryotic genes. Then we have bacterial artificial chromosomes that are derived from F1 plasmids. They are capable of carrying the DNA up to 300 kilobase pair. So these are the plasmids which we have used. Plasmids, not plasmids, these are the vectors which we used. The plasmid, bacteriophage, cosmid, this yeast, this expression vector which will use to carry the eukaryotic genes and also helpful of doing the transcription and translation inside it. Then we have this yeast artificial chromosomes, we have bacterial artificial chromosomes. So these are the vectors which are used in the recombinant DNA technology. Now we have the techniques which are used in this RDNA technology. Gel electrophoresis, we have done cloning libraries, enzyme mapping, PCR, nucleic acid hybridization, and DNA microarrays. These are the, you can say, different types of techniques which are used in recombinant DNA technology. Then we have some applications of RDNA technology. First of all, let me check whether you get it. So are you able to get it? So before moving forward to the applications, I will show you one video animation. So you are able to get it. Come on, reply. Be active, please be active. We have Salil here. Who is Salil? Salil, how are you? Are you new in this lecture series? Is this your first lecture? Salil, from where you are? I just read your name today only. So how are you, and is it your first lecture today? Sleel? No. Okay. Yes, we have Amita here, Vishakha here. Okay, good, good. So Sleel? Yes, yes. How are you, and from where you are? You are also from our Khalsa college. Kindly let me know. Vrinda is here. Okay, Namrita is also here. Okay, okay, good, 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 good. So are you able to get it? Okay, you are third year. Okay, good, good, Salil. So if you have any type of, if some issues are not clear or some topics are left, then kindly let me know. We will discuss them also. Okay. So let me show you one small PPT on it. But I don't know which one is the recombinant technology. Let me check it.
Welcome back,
No,
guys.
this is
In this
Liza.
video,
1 minute. I have one video of this recombinant.
or enzyme linked immunity.
I also have to share all these
Welcome to
PPTs.
biology exams for
Yes,
recombinant.
this is our
Today we
DNA.
are going
Okay.
to discuss in detail about the steps in recombinant DNA technology. First of all, we will start with definition. In this video, we will be going through all the processes in detail. Recombinant DNA technology is a technique that is used to refer identification, isolation, and insertion of gene of interest into a vector such as plasmid or bacteriophage to form a recombinant DNA molecule and production of large quantities of that gene fragment or product encoded by that gene. We'll be going through different steps in detail in this presentation. First of all, let us have a look into the synopsis of the steps in recombinant DNA technology. Step 1. It is the identification and isolation of gene of interest or DNA fragment to be cloned. Second, we have to introduce this isolated gene into a suitable vector. Then we need to introduce this vector into a suitable host. The process is called as transformation. We need to select the transformed host cell, and finally our intention, multiplication or expression of the introduced gene inside the host. So let us move into the first step, that is identification and isolation of gene of interest. In recombinant DNA technology, from where we get this gene of interest, there are different resources for getting our gene of interest. These include genomic library, cDNA library, chemical synthesis of a gene if we know the sequence. If the number of copies of a particular gene is very less, not sufficient for gene cloning, we can amplify that using polymerase chain reaction, gene amplification technique. So in order to understand all this concept, let us take an example. Here we are going to introduce human insulin gene in bacteria. Step 1. From genomic library, we have taken human insulin gene. As I mentioned, genomic library is a source from where we get our gene of interest. So step 1 is I have taken human insulin gene from genomic library. Step 2, I need to insert this desired gene, that is the insulin gene, into a suitable vector. Before that, we need to know something about vector. What is a vector? Vector is any DNA molecule that has the ability to replicate inside the host to which our gene of interest has integrated for cloning. Vectors include plasmids, bacteriophage, bacteriac, etc. You can refer our video gene cloning vectors for more. So most often here we are taking plasmid as our vector. Plasmid is extra chromosomal DNA that is present in bacteria. This is E. coli bacterium. I have isolated plasmid from E. coli bacterium. This is a plasmid vector. So this is my gene of interest. This is our gene of interest, that is a human insulin gene. So first of all, we need to make a cut in the vector using a restriction enzyme. Restriction enzymes are enzymes that are capable of making internal cuts at specific sites in a DNA molecule. So we need to incorporate our insulin gene into this vector. So what we did was I made a cut using restriction enzyme, and now I have the gene. So I have incorporated my insulin gene into the vector, and I seal that gene, the cutting ends with the enzyme ligase. Now we have the recombinant DNA molecule or chimeric DNA. A recombinant DNA molecule is a DNA molecule from which DNA are from different sources. You can see right here we have a recombinant DNA molecule with human DNA and bacterial plasmid DNA mixed together. So just recombinant DNA molecule is just a recombined DNA molecule from different organisms. So second step is over. We have inserted our human insulin gene into a vector. Now the third step, introduction of this vector into a suitable host. There are different gene transfer methods by which we can introduce our vector into a host. These are some of the methods: electroporation, microinjection, liposome-mediated gene transfer, silicon carbide fiber method, etc. And hopefully we'll be discussing all these later. And chemical gene transfer methods are there: PEG-mediated gene transfer, calcium chloride-mediated gene transfer, extran-mediated gene transfer, or certain cells like bacteria are capable of taking up gene from the surroundings. The process is called as transformation. So now our insulin gene, we have our insulin gene incorporated in the vector. So we need to introduce this insulin gene into a host. Most often the host is a bacterial cell, as it is simple to manipulate, and we know E. coli bacterium very well. So I have introduced my vector with my gene of interest into bacterial cell. Now we have a genetically modified E. coli with our insulin gene inserted inside the vector. So step 3 is over. The process is called as transformation. Now step 4, after this step 3, we will be getting 3 types of colonies: non-transformed bacterial cell without any change, and transformed bacterial cell with unaltered vector. Vector is there, the cell has transformed but without our gene of interest, and finally the transformed bacterial cell with our recombinant vector. And this is the cell that we need to take, and this is the colony we need to select, and this is we need to select this particular colony from the rest. Majority of the colonies belongs to these 2 groups. So there are different ways by which we could select these, maybe by growing in antibiotic resistance in a selective medium, or this gene may be coding for some characters that are visible, or maybe if it is an enzyme, we will be adding some substrate, we will be getting some color in some colonies in which we will be having our gene of interest, maybe by Golding hybridization, clotting test, etc. For antibiotic resistance selection in recombinant selective medium, you can refer our video gene cloning vectors. Final step is multiplication or expression of the introduced gene in the host. So as I mentioned in the first slide, our intention is to make as many copies of that particular gene, or I need to make that gene product to be synthesized inside the bacterium. So we have genetically modified E. coli with our insulin gene, and these plasmid will replicate inside the host, and these bacteria will also replicate, making millions of copies of this particular gene. Our first intention is achieved. We have our gene of interest produced in millions of numbers. Our intention was to synthesize insulin inside E. coli. So this is what is happening. This E. coli gene that will be transcribed and translated later, protein product is synthesized inside the bacterium. So expression, that is the term, that is insulin is synthesized in bacterium. Now by recombinant DNA technology, insulin is synthesized. The name of that insulin is called as humulin. Humulin is synthesized by recombinant technology. Here we are using the same procedure. We are making bacteria as big biofactories for synthesizing human insulin inside. Yeah, and so we need to culture these in large scale for getting large scale production of insulin. Hope everything is clear.
So yes, this is a small video on the basics of recombinant DNA technology. So is it clear to all of you? Okay, is it clear? Shall we move further? Kindly let me know. Shall we move further? There are some applications of recombinant DNA technology which we have to cover. So kindly let me know. Okay. Good. Okay. Now we will move further with our, these applications of recombinant DNA technologies. Okay. So, so we have done these are another techniques which are there in the rDNA technology, but my question is same. If we have PCR here for amplification of a particular gene segment or a DNA segment, then why we need rDNA technology? So it's my humble request to all of you that kindly go through it and let me know tomorrow. Then now we will move further to our applications of rDNA technologies, that is why this, this broad technology or rDNA, why this technology is so prominent, or why it is used so much. So basically the applications are manufacture of protein, hormone, interferon, plasminogen activating fiber, blood clotting factors, insulin, so growth hormone. Basically they are saying that if we want to extract insulin gene, so for this we need this technology. If we want to isolate growth hormone for the growth of a children, like there are some children who are not going so fast, so growth hormone for the isolation or for the multiplication of growth hormone we need rDNA technology. For AIDS test also this technology is used. Then diagnosis of molecular diseases like sickle cell anemia, in which the RBCs become sickle shaped. Then we have this disease, we have cystic fibrosis. So for these all we need the rDNA technology. Then we have gene therapy. This is achieved by cloning a gene into a vector that will, they are telling us what is gene therapy basically. So this is achieved by cloning a gene into a vector that will readily be taken and incorporated into the genome of a host cell. So first we have a vector along with our gene of interest, which has to be taken into the host cell. So like this is if bacteria is a host cell, so bacteria has its own gene, but our plasmid comes inside with our desired sequence or desired segment. But when this plasmid gets incorporated into the nucleus or into the DNA of the bacteria, incorporated into it is known as gene therapy. So they have also applications in agriculture, like genetically engineered plants. We have so many genetically engineered plants or crops these days, like we have a apple and we have a brinjal which is genetically modified. So for those purposes also we need a rDNA technology. Then some industrial applications, that is to produce sugar, cheese, detergents, or some type of food additives, or some type of another foods by adding so much of flavor. So for all these purpose we need this technology. Yes, this technology is very important in forensic medicine, or you can say in forensic science. So the restriction and analysis pattern of DNA of one individual will be specific, but the pattern will be different from person to person. Helps to identify criminals and to settle the disputes of parenthood. So for forensic purposes also this technology is used. Even PCR is used for the forensic purposes also. So these are some of points where this technique is used. Now we have another term which is gene cloning. Cloning is basically what? Cloning is basically the replica of the one thing. That's a clone. Like we have a dolly sheep which is a clone basically. So that's the exact copy of a one segment, or you can say exact copy of the one thing or one organism. So this is gene cloning. I will tell you one difference. In PCR, basically we said there is an amplification of a gene. In PCR also there is a multiplication of the genes or the DNA segments. But in rDNA technology, we said it as gene cloning. What we said in case of rDNA technology, it is known as the amplification. Amplification is what? Multiple copies of a single gene segment or a single DNA segment. But in case of rDNA, instead of saying amplification or multiplication, it is known as cloning. So what is basically the gene cloning? The recombinant DNA molecule is transferred to a host cell. So first of all, the recombinant DNA, that's a plasmid in a DNA, is transferred into a host cell. Within the host cell it replicates. Within the host cell it replicates, or you can say it amplifies, it multiplies, and it is known as cloning. So it is known as when we are able to make identical copies. So this is the one difference in case of PCR or you can say rDNA technology. In PCR it is known as amplification or multiplication, but in case of rDNA it is known as gene cloning. So the cloned DNA can be recovered from the host. This is a property of the host, that the cloned DNA or the multiplied DNA can be easily passed outside again, or it's also a property of a plasmid DNA. So gene products can be isolated and used for research or for some other purposes. So this is the cloning, or you can say multiplication of the particular DNA segment. As usual, we have a site of interest which we want to, you can say, which we want to multiply. Like suppose this is for the AIDS detection, you can say, for example, or you can say for insulin for diabetes. Insulin, okay, suppose. So this DNA segment gets inserted into the plasmid. Now this recombinant plasmid, also known as chimeric plasmid, is inserted into the bacterial cell. Okay. Now this bacterial cell has its own chromosome. Now what happens? There is a multiplication, or you can say amplification of the rDNA. So if you are able to see here, we are able to extract the plasmids, and we are able to extract this particular segment. If you are able to see here, so these are only these segments. Here they are 2 and here they are 6. So we are able to clone or multiply this particular DNA segment. Okay. And what happened on this side? To obtain protein. So what happened here? Grow under conditions that allow expression of a cloned gene. Now what happened here? There is a mixing of the, like this is the characteristic of gene therapy. So there is a mixing of the two DNA or chromosomes, and there is a isolation of the protein. So this is the whole visualization of rDNA technology, the whole visualization that how this process operates basically. So if you are able to see here, this plasmid contain antibiotic resistance gene. We had our DNA of interest, then again the replication, or not the replication, again the amplification, or you can say cloning occurs here. Okay. So this is all about rDNA technology. Jelly electrophoresis we have done before, PCR we have done before. So these are applications. I think I have one more PPT on applications. Let me check. I must have. Yes, I have. So let's discuss the properties of our recombinant DNA technology in more detail, because it's a special topic in your lecture series also. So I will elaborate it more. 1 minute please. 1 minute please.
Process of recombination of plant or
Yes.
DNA
This
technology.
is also one video. I think we have not seen it yet. Just see it and then we will start the applications of recombinant technology. I think
1.
there must be applications.
Isolation of genetic material. The first step is the isolation of desired gene from a cell and the digestion of bacterial cell wall by enzymatic action. This is achieved by treating the bacterial cells of plant or animal tissue with enzymes like lysozyme bacteria, cellulase, plant cells, and chitinase, fungus. Genes are located on DNA molecules centrifugation with proteins, such as histones. RNA can be removed by treatment with ribonuclease. Proteins can be removed by protease. Other molecules can be removed by appropriate treatments and further subjected to gradient antifugation. Ultimately, purified DNA is precipitated out after the addition of chilled S2. Cutting of DNA at specific locations. The purified DNA is cut into number of fragments by enzyme called restriction endonucleases. Each restriction endonuclease functions by inspecting the length of DNA and recognizes a specific palindromic nucleotide sequence in that DNA. Then it will bind to the DNA and cut each of the two strands of the double helix at specific points. These are called molecular scissors. They were first discovered by Nathans in 1970. Over 250 restriction enzymes have been isolated so far. When linear DNA is treated with restriction enzyme, a large number of DNA fragments are formed. The resultant fragments are separated by gel electrophoresis. Finally, the desired DNA fragments are selected by Southern blotting technique. 3. Amplification of gene of interest using PCR. Once the desired fragments of DNA genes are obtained, they are inserted into a suitable vector to produce indefinite number of copies of genes. This is known as gene cloning. A cloning vector acts as a vehicle to carry the desired gene. An ideal cloning vector should have following properties. 1. It must have low molecular weight. 2. It must have unique cleavage site for the activity of restriction enzymes at single point. 3. It must be able to replicate truly inside host cell after its introduction. 4. It must contain genes which provide resistance to antibiotics. Several types of vectors are used in recombinant DNA technology like plasmids, phages, etc. The enzyme DNA polymerase helps in replication. If the process of replication of DNA is repeated many times, the DNA segment can be amplified to approximately billion times. That is, 1 billion copies are made. Such repeated replication is achieved by use of thermostable DNA polymerase, which remains active during high temperature induced denaturation of double-stranded DNA. The amplified fragment of desired gene can be used to ligate with vector for further cloning. To isolate a plasmid, the bacterial cell is treated with EDTA, ethylene dimine tetraacetic acid, along with lysozyme enzyme to digest the cell wall. Then the bacterial cell subjected to centrifugation in sodium lauryl sulfate solution to separate the plasmid. The isolated plasmid DNA is cut with same restriction endonuclease enzyme. The enzymatic cleavage cuts circular plasmid into a linear molecule having sticky ends. The two sticky ends of this linear plasmid are joined to the ends of the desired gene. The enzyme DNA ligase joins the complementary ends of plasmid DNA with that of desired gene by covalent bonding to regenerate circular hybrid called recombinant rDNA or chimeric DNA. 4. Introduction of recombinant DNA into the host. The recombinant plasmid is transferred into a suitable bacterial host cell, generally E. coli, by a method known as transformation for the expression of desired gene. The cells into which the recombinant DNA are inserted are called transformed cells. Example, if rDNA bearing gene for resistance to an antibiotic, example ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin-resistant cells. Only transformants will grow on agar plates containing ampicillin. The transformed recipient cells will die. The ampicillin resistance gene is called selectable marker. Bacterial cell walls are not ordinarily permeable to such recombinant vectors, but reaping in dilute solution of calcium chloride renders the bacterial cell wall permeable to the recombinant vectors. Inside the host cell, this rDNA starts replicating. The transformed cell will begin to grow on medium and divide as separate units. The replicate vectors in each cell are passed on to daughter cells, giving rise to clones.
Basically, this is all regarding the recombinant DNA technology which we have discussed today. Now we have some another applications of recombinant technology. So should we discuss it now? Are you comfortable? Are you not get bored or tired? Everything is fine? Today we have a good connection also. This is another thing, that there is not any type of buffering. So like sometimes there is buffering also, which causes lots of trouble. So everything is okay. So we have only just small topic of the applications of recombinant DNA technology. Let's just do it, and then we will conclude this recombinant DNA technology. So yes, they are showing thumbs up. That means they are all are fine. They are not getting bored. That's very good thing. Okay, good, very good, very good. And also let me know whether this timing of 4 p.m. is okay for you. Because on the 4 p.m. there is no noise, even on the outside. There is so much peace in the outside everywhere. So is the timing of 4 p.m. is okay to all of you or not? Like for tomorrow, are you free on 4 p.m. or not? Because I found this timing okay. There is not even a problem in internet, or internet connection is also good at that time. So kindly let me know if you are comfortable to proceed the section or the lecture on 4 p.m. tomorrow. Okay, so yes, they are just saying yes, ma'am. I'm just waiting. Okay, so Jiveria, are you free, or is there any practical on tomorrow also? Please let me know either here, or even you can WhatsApp me. So if we have practical tomorrow. Okay, then we will start at 4:30 or 5. No issues at all, because I will be there by 4:30. Obviously we chat for 5 or 10 minutes. Then we will. Okay, no issues at all. No issues. Just go with your practicals first. Okay, dear. Okay. No issues. So just go with your practicals first, then we will take our section. No issues at all. Okay. So okay. Okay, dear. Okay. We will start by 5. Basically, I will be there, so that there should not be any type of problem on this online or offline connection. So you just join after your practicals. There is no issues regarding it. Okay. Okay, no issues, because practicals are also important. Okay, now I'm just going with the applications now. Ma'am, well, when will be the last lecture? I will tell you, just let me finish this applications of RDNA, then we will talk about it, okay? And also you are here, please like the video, only two like the video. Are you not liking recombinant DNA technology? Please like it. It's just an appreciation from your side, so please like it. Okay, now I will talk later, let me finish applications first. Okay, so now we have only one thing, which is the applications of recombinant DNA technology. Obviously we have done it in the PPT also, so but let's once again revise it. I think it's not visible to you, maybe. 1 minute. Yes, okay. So the number one application of this recombinant DNA technology is in the field of health. Like we have discussed from the beginning, that only the particular DNA segment is amplified, or you can say cloned, which is specifically for any disease, or which is for curing any type of disease, or even there are some type of growth hormones on which we are working. So basically the number one is production of vaccines. So vaccines are live attenuated and modified pathogens given to an individual to trigger the immunity. So basically this R recombinant DNA technology is also used, or is used for the purpose of production of vaccines. So I don't know whether you are seeing it or not. I will share this PDF with you. So by recombinant DNA technology, antigen producing gene and plasmids combined, and then rDNA formed and used as a vaccine. So basically the vaccines are generally for HIV, hepatitis B, then we have herpes, mouth and foot diseases. For these purposes we are using the rDNA technology. Then we also use it for human growth hormone, like I told you before, that's for growth, regeneration, or for the activities of the humans. We are using the recombinant DNA technology. Then monoclonal antibodies, or the artificial synthesis of the antibodies. So artificially prepared antibodies available by rDNA technology, and they denature harmful living pathogens, and the example of which is penicillin. So we have you can say artificially prepared antibody, which is penicillin, which is made in 1920 by Fleming. So for the production of antibodies to work against to work against antigens, we have the specific antibodies, or you can say artificially synthesized antibodies. So on which we have one topic, which is monoclonal antibodies. It is a production of antibody occur in the presence of specific or foreign antigen particles. So our blood contains several antibody serum derived from different antigenic molecule. Further, with isolating culturing procedure, these specific lymphocytes are cultured in vitro, known as monoclonal antibodies. I have to show you the applications of this monoclonal antibody. Basically, it is used as diagnostic agent of disease, infection, and also used in pregnancy test. It is also used in the treatment of cancer and veterinary cases, and develop effective vaccines for human, animal, and plants also. So these all are things which are used, or which are in which you can say recombinant DNA technology is quite helpful. Then we have to do molecular diagnosis of disease. For this we need recombinant DNA technology. Then we have gene therapy, which we have discussed before also. DNA fingerprinting, which is also known as forensic medicine. So for all these purposes we are using the recombinant DNA technology. I am not reading it thoroughly because I will share it with you. Then we have applications of recombinant DNA technology in agriculture, like we have discussed genetically modified crops like brinjal, like apple. For those things we want to production of stress tolerant crops, we want to increase the yield of crop, and we want to enhance the variety of crop. For those purposes also we need the recombinant DNA technology. I don't know why I don't know either we need it or not, but they are using this technique for the enhancement or for the increasing the size of a fruit, for making it beautiful or attractive, and also to make it disease free or pest free. For these purposes we are using the recombinant DNA technology. So, and also we are able to generate or produce hybrid plants which are herbicide tolerant, which are not attacked by any type of infection or pest. Then insect tolerant plants are there, like not basically insect, you can say pest, the those type of insects which are harmful for the plants. So we have insect tolerant plants, then we have stress tolerant plants. So these all are the applications of recombinant DNA technology to increasing quality and yield of crop. So with the help of this technology we are able to enhance the yield of our crop, either wheat or maize or any type of crop. With the help of it we are able to enhance its yield. Then production of biopharmaceuticals, that is in the industrial field, they are using this rDNA technology. So these are the applications of rDNA technology. That is, it is used in food industry, it is used as beverage industry or fermentation, agriculture industry, they are using it. I will share this PDF, even I will share all these, you can say, the topics which we have, the PPTs I will share, and then you will just read the things and kindly let me know if there is any type of problem, or you are not able to get it. So I think that's all from my side for today, that is recombinant DNA technology along with applications. So kindly go through it. If there is any topic or if there is any type of query, you can also let me know. Now you can ask me any question regarding this. Yes, Devita, actually I forget always. Kindly share your mail ID here, kindly share. If you are able to share your mail ID, please kindly share email ID. Jaguriti, kindly share the email ID. And kindly like the video, only 7 persons like it. Why are you not getting my points? Is it not a good lecture today? Is it on? Yes, it's on. Hello. So kindly you share your email IDs, please. Okay, Jeet. Yes. Kindly share your email IDs, please. Yes. Okay, Jiviri, I have your email ID. I know. So if any other one is there who is interested in these PPTs, kindly let me know, or even your friends will share with you. Kindly reply please. Manpreet, Devita, okay. So kindly reply. Are you able to get our DNA technology? Are you feeling it okay? Everything is clear to you? So kindly reply. Why are you not replying? Yes, Juvelia is asking, will okay, Juvelia is asking something. I just can send the PPT. Yes, when we will be the last lecture. Actually, we have 2 topics left, which one student has shared with me today. The very first, which is interaction regarding MBBT, and we have cell culture techniques. So I will try to cover it today. If it's not possible to cover it today, not today, tomorrow, I will try to cover the topic on tomorrow, that is on Thursday. If it is covered on Thursday, it's okay. Otherwise we will continue it up to Friday. So all are you comfortable in it? Kindly reply that we have 2 small topics left. Either I will try to cover it tomorrow or the next day after tomorrow. So if you kindly share your thoughts with me, then it will be nice, all of you. We have Jeet, is it okay? Jeet, where are you? Okay, Manpreet is here. So as only few days are left with all of you, so I want to say all the best to all of you for your future projects, for your practicals, for your assignments, and please be in touch with me and always be there along in my another video sessions also. Okay, good. Julia says it's okay. Okay, very good, dear. I'm thinking that you have your exams maybe. That's why I'm asking. So we are here for this week. We are together for this weekend, for the up to Friday or Saturday maybe. So, but I want to say you that please be in touch with me, because you are very nice students and very cooperative also. So I want to thank all of you. Really want to thank all of you. So I request you to all be in touch with me. Okay, dear, I will take. Okay, Jeet, I will take bioinformatics also. No issues at all. No issues. Actually, I want to do bioinformatics in quite detail, but I will do the basics bioinformatics. I will definitely cover. You need not to have worry. So just be in touch with me, and there will be lots of other videos will come. So you have to learn something good from those videos also. So is it okay for today? Is the session is okay for today? Everything is fine to all, is fine to you? Yes, please, where is Priya? Kindly reply. Where is Rituja? Where is you? We have Ankita. Where is Salil? The person whose name I just read today. Where are you, Salil? We have Nupur. Where are you, Nupur? Bhagyashri, Gayatri. Are you here or not? Siddhi, Cloris, Shubh. Where are you all? Amita, Vishakha, where are you all? Kindly reply, please. Please reply. Debita is here. So I am waiting your messages only. By the help of these messages, I am able to know whether you are getting it or not. Okay. Yes, they are here. Nupur is here. Rutuja is here. Salil is also replying. Very good. Jagriti is here. Gayatri is here. Thank you, Gayatri. Thank you so much. So I just want to say keep in touch. Okay. So all the best for your tomorrow's practical, Juveria. Do good. And also the other batch, I'm sorry, I don't know who are there in the batch of Juveria. So my best wishes are there with all of you. And also kindly like the video, okay? And my best wishes are with you. We will meet tomorrow. Okay. So we will meet tomorrow with good positive vibes, and we will cover the only topics which are left in this session or in the lecture series. So that's all from my side. Thank you all. Thank you very much. We will meet tomorrow. Yes, Vishakha, thank you. Thank you, Repita, thank you. Okay, Ankita is saying yes, ma'am, always. We will meet at 8. Yes, definitely, Jeet. Kindly support me in the biochemistry section, because the students are, I don't know, they are from different streams, not from different streams, basically, they are from different areas, so they have some different opinions regarding everything. So I hope you are my MBBT students. You will definitely support me there, and of course you support me on the last session also. So I want to thank all of you who were there even the whole time. So I want to thank all of you also. Okay, I will not skip. But there is only one girl who is saying that ma'am teaches too basic. I said this is the thing in the topic, so I have to cover it. So that is why, otherwise I have to start from. Oh, no, I will not skip. Definitely. Why should I skip? When I have a proper, you can say I have a proper topic given to me, why should I skip? This MBBT series is actually given to me later on. You all know regarding it. You have some another teacher who is guiding you maybe or teaching you. So that's why some topics are left in this session. But on the biochemistry session we will complete everything. Either we will take it, either we will cover it in 10 days or in 12 days. It's the different issue, but we will cover everything or every topic. Okay, so you know you need not to worry. We will definitely cover. Okay, yes, definitely. No, ma'am, we need basics as well. Yes. Tomorrow in tomorrow's not in tomorrow in today's biochemistry section, we have to study hormones, and it's also very interesting topic for you, for all of you. So we have to study steroid hormones, we have to study protein hormones, and one topic, that is central dogma, is left on that session. So we have to start with central dogma, then we will move forward towards hormones, steroid hormones, protein hormones. Okay, so that's all from my side. We will meet by 8 p.m. So till then, take care of yourself and all the best for your practicals. Okay, bye. So should I end stream it now? Yes. So okay, bye bye. We will meet again. Till then, take care of yourself. Thank you. Bye. Jiveria, bye. So that's all from my side today. We will meet tomorrow. Okay, so that's all. Bye. Take care of yourself. Bye. Bye Ankita. Bye Gayatri.